My name's Aridgides. I'm a pulmonologist at Dartmouth, and I'm a member of the RCMB Web Committee. And today I'm delighted to host Viral Shah from MGH, who's going to be presenting his journal club on a recent publication titled Airway Hillocks are injury-resistant reservoirs of unique plastic stem cells. And we are excited to have several panelists today to help provide their perspective on his work. We have Lida Hariri, who is one of the collaborators and a pathologist at MGH, along with Viral. We have Jayaraj Rajagopal, who is the PI of the work. And then we also have a couple of external people to provide their perspective. Alan Fine from BU, who is another expert on pulmonary stem cells, and then Erin Crossey, who is a co-member of mine on the web committee and works with Alan. Just to remind everyone, the session will be recorded and posted on the ATS website, and a recording link will be sent to all registrants. We're going to give the first part of the session to Dr. Shah to present his work, and then I'm going to ask all of the panelists to give a few minutes of perspective on what they think was interesting and important about this work, and then we'll open it up to questions. Dr. Shah has indicated that he's also open to being interrupted during the course of the talk, if you'd like. I'll monitor the chat and raised hands if you'd like to do that and serve as a moderator in that fashion. So with that, please take it away, Viral.
Great. Well, thank you so much, Dan, for inviting us to share our work here, and for the ATS for also inviting us and setting all this up. We're really excited about this recent manuscript and love to share with everyone. Hopefully we can make this more of a discussion rather than me just presenting everything at you. You can just read the paper if you just want that. So please, let's have this interactive. I only have about 30 slides. So we'll have plenty of time for discussion. So if something's not clear, feel free to stop me, or if you want to add an anecdote, or panelists, if you want to interject, please feel free. So today I'll give you a little bit of background and a little bit of perspective on kind of what led us to this work. Then I'll talk a little bit about the mouse, our findings of defining hillock properties in the mouse. I'll talk about defining hillock properties in human airways. Kind of put this all together with implications of this work and the next ongoing steps and ongoing work. And I'll summarize the main points of our findings and have a nice illustration, a cartoon of how we put this all together. All right, let's get going. So for a long time, this is what we thought the airway epithelia was. We thought that it's a pseudostratified epithelium, meaning all these cells touch the basement membrane. And sometimes they may not look like they do, but they all do. They're pseudostratified. And there's three main cell types. There are secretory cells, there are basal cells, and there are ciliated cells. And then in 2018, out of the Rajagopal lab, this paper Montero-Haber Bidon et al. Changed the way we think about it. Definitely changed the way I think about the airway epithelia. And that it's a little bit more complicated than that. And so this was through single-cell RNA sequencing. It revised the airway epithelial hierarchy. And that there's different flavors of these common cell types. And it identified that there's actually different types of rare cells, and specifically identified the CFTR-rich ionocyte. And I was really excited because this cell had the most CFTR out of all the other cell types on a per cell basis, and clearly had very defined or very exciting implications for disease. And there's a lot of ongoing work trying to get at that. But also in this paper, they also described this cell called the hillock. And that's what we're going to focus on today, the hillock. And initially, the role of the hillock, its implications, what it wasn't totally clear. And the ionocyte was the exciting part of that paper. It's literally in the title. But today, we're going to talk about what we found and what we've learned about the hillock. So to get started, hillocks aren't just a single cell. Hillocks are unique structures. And so let me orient you on how I'm going to show a trachea explant in this study and in the paper. So this is a tracheal explant you see here on the right side of the screen. We essentially dissect out a trachea from a mouse. That's what you see down here, the bottom of the screen. We cut along the anterior aspect of the trachea and unroll it so it's flat. And so that's what you see here. The lateral sides are actually the anterior aspect of the trachea, just cut and unrolled. Here at the center, you have the posterior membrane, and then you can see the cartilage rings on the sides. What we found was that hillocks are in characteristic locations on the mouse trachea. They're over these cartilage rings and along the posterior membrane and some of the adjoining regions between the two. And what you see here on this image is expression of keratin 13. And we found that hillocks uniquely express keratin 13, unlike all the other cells in the murine airway. And this is important. It's in the murine airway. Keratin 13 is unique. Hillocks also have a stratified squamous structure. And so here's a cross-sectional image of this. We have acetylated tubulin in magenta that stains ciliated cells, keratin 13 in green, and DAPI in blue. And what we see here is surrounding the hillock at these two ends, we see ciliated cells. So ciliated cells are outside the hillock. And in the hillock itself, we see that the cells highly express keratin 13. That's this green staining here. And in fact, there's two layers of cells here. There's a luminal layer that highly expresses keratin 13, and also a basal cell layer, as you can see down here, also expresses keratin 13, but slightly lower than the luminal cells. Now, things can look stratified, but may not be. So we wanted to be very rigorous in defining if hillocks are truly stratified. So to get at that, we used a photoactivatable GFP. And so I'll explain this to you in a second. But we express the photoactivatable GFP across all the epithelial cells. And so when we stimulate with a certain wavelength of light, it activates the GFP. And so the certain cells will glow green. Now we can do this on a single cell basis. And so we decided to do this for hillocks and other pseudostratified epithelia. And so this is what you see here on the right side of the screen. So PA-GFP, photoactivatable GFP, here in green, and second harmonic generation, which is autofluorescence of collagen in gray. And that denotes the basement membrane. And you see secretory cells here, their characteristic shape in green, and they touch the basement membrane. So they're pseudostratified. When we look here at the hillock, specifically a hillock luminal cell, we see the hillock here is in green, and the basement membrane is SHG in gray. There's this space between the two. It looks like the hillock is actually floating. And it's not floating. There's actually multiple cell layers here. And so this suggests the hillock is truly stratified. And so here's a video of two different hillock luminal cells where you can see the SHG staining the base or autofluorescence of the basement membrane is definitely not touching the hillock luminal cells. And so hillocks are truly stratified structures. And that's surprising. We don't expect the airway to have stratified structures. Other organs, like the skin being one of them, that tends to be stratified. So we then wondered, are there other properties of the hillock that also could resemble skin? And so here we looked at, here's SEM, looking at pseudostratified epithelia and hillock. And we can see in the pseudostratified, we have ciliated cells, the beautiful cilia, and secretory cells. You can see here on SEM. The hillock looks very different. There are no ciliated cells. There's no secretory cells that we see here. It's very flat. And you can see these very thick boundaries between the cells. I like to think this is like the arid desert, and here we have the pseudostratified jungle, right? They're very different environments. In fact, these boundaries are really important in the hillock. Now looking at TEM of a cell-cell boundary between the hillocks, we see that they're enriched in desmosomes. And that's also very reminiscent of the skin. Skin highly expresses desmosomes and intermediate filaments. And so we can look at gene expression, and this is reflected in the gene expression data here, looking at expression level on the y-axis for hillock luminal cells and hillock basal cells compared to pseudostratified basal cells. we see that the hillock highly expresses keratin 13. That's, as I've shown you before, as we would expect, but it also highly expresses desmosome proteins. This is desmoglein 3, and this is uniquely expressed here in the hillock. And here's immunofluorescence also demonstrating this, with keratin 13 staining in magenta, desmoglein 3 staining here in green between the cells. And then looking at the pseudostratified epithelia denoted by ciliated cells here, we don't see any of that desmoglein 3 or keratin 13 staining. So hillocks resemble skin in being stratified, and some of their properties here as well, desmosomes and intermediate filaments. Well, the skin plays an important role in protecting us from the environment, right? It's a barrier structure. So we then wondered, would hillocks also serve as a barrier in the airway, a protective function in the airway? As we know, the airway is exposed to the environment constantly and needs to repel the environmental insults. One of these insults, and this is what I tell the pulmonary fellows all the time, is that everybody aspirates. Gastric contents, something that always comes up into the airway and can cause havoc. And so we wondered, would hillocks survive acid injury? So we took a trachea, dunked it in 80 millimolar HCl, and then stained for dead cells here in magenta, and hillock cells, keratin 13 specifically in green. And what you can see, the keratin 13 cells, the hillock here is at the center, and the majority of the dead cells are surrounding the hillock in the surrounding pseudostratified epithelia. So we can quantify this looking at dead cells per millimeter squared here on the y-axis for pseudostratified cells compared to hillock. And the hillock, there's less dead cells. So this is one injury that the hillock seems to be protected from. Another very common injury or insult that we're exposed to, that we're all familiar with, are viral infections. One of the most common ones is influenza. So we then asked, is the hillock protected from influenza? So here we see again keratin 13 staying in the hillock in green, influenza nuclear protein here in magenta, and we see that the flu is excluded from the hillock. Purely the pseudostratified epithelia is what gets infected. We can quantify this here on the y-axis, percent of the surface area that has flu nuclear protein. Pseudostratified compared to hillock. Hillock is protected from flu. We then wanted to look at what else could the hillock survive. And one of the things, when I was on my interventional pulmonary rotation, one thing we do, right, is use cryo. We use cryo to freeze part of the airway. So I was wondering, does the airway survive after cryo? Could hillock survive that? Actually, similarly, Jayaraj Rajagopal, my mentor, when he was a postdoc, he was trying to decellularize trachea. And he would stick it in the -80 without cryoprotecting. Presumably, everything would die. But he noticed some cells survive. So together, we kind of put this together and we wondered, do hillocks survive cryo injury? So we literally did both those experiments. We sprayed the trachea with liquid nitrogen. We also stuck it in the -80 twice, and then stained with a dead stain here in magenta, and stained for hillocks and keratin 13 in green. And what's pretty striking here is that the epithelia surrounding the hillock, the pseudostratified epithelia, is primarily dead, while there's very few dead cells in the hillock itself. And we can quantify that again here on the y-axis, dead cells per millimeter squared, pseudostratified compared to hillock, and the hillock's protected. So the hillock survives a panoply of different types of injuries, from HCl, influenza, cryo injury, and I'll talk about one more in a second, which is naphthalene toxin. But what does it do after injury? And it turns out the hillocks not only survive injury, but then they in turn reepithelialize the airway surface. And so here's hillocks denoted in green. This is a mouse that's exposed to naphthalene smoke toxin, which kills a number of the pseudostratified epithelia. And you can see over live imaging across 5 days that this hillock region expands across the airway surface. And so we wondered, what's going on here? Are the cells just spreading out? Or is there actual basal cell replication and regeneration of the airway surface? And so to get at that, we had to use a specific basal cell driver. So in this next experiment, we used a P63 CRE-ER, so all basal cells, and we used a mosaic mouse, a mosaic reporter. And this essentially means that all the basal cells that are labeled will each cell will be a different color. And so this will allow us to identify different clones of basal cells. So we repeated that experiment that I just showed, this time 3 weeks of, after 3 weeks of injury, we then explanted the trachea and saw what happened to the basal cell clones. So just as a reminder, this is the location where we would see the hillocks, along the posterior membrane and the cartilage rings. And what we see with the clonal resurfacing is that there's these massive streams of cells that come out of the hillock regions. We see these streams of cells coming out from the posterior membrane and from around the cartilage, hillock-specific regions. We can take a closer look at these, and remember, each color is a specific stem cell basal cell clone. And we see thousands of cells, all derived from the same clone. And we can quantify this, and that's actually, we see tens of thousands of cells coming from single basal cell stem cell clones from the hillock. Now, again, I mentioned we wanted to focus on basal cells, and this was a basal cell driver. What we wanted to do is be more specific for the hillock. And so what we next did was develop a hillock lineage mouse. So as the hillock is the only epithelial cells that express keratin 13, we use this as our driver. And so we knocked in CRE-ERT2 into the endogenous KRT13 locus to generate a KRT13 CRE-ER mouse. Here is with a reporter, LSL-TD tomato. And so we looked here to see whether hillocks would express TD tomato. We stained for keratin 13 protein in green, and you see a ciliated tubulin here, which is a surrounding sort of stratified ciliated epithelia around here in blue. And what you see is our driver is pretty good. Looking at the efficiency and specificity, it's pretty high in labeling the hillock cells and only the hillock cells. So now we can label hillock cells. So we repeated that experiment. This time, we labeled hillocks. We waited a week for washout, and then we performed naphthalene injury. And then we looked to see what part of the epithelia was derived from hillock. And you can see hillock lineage in TD tomato in magenta here. K13 staining, which is staining the present hillock here in green, and a ciliated tubulin staining the ciliated cells in blue. And we see 3 weeks after naphthalene injury, there is a dramatic resurfacing of the epithelia with hillock lineage. So the hillock expands and resurfaces the epithelia. Now, I just mentioned the hillock is squamous. So is it resurfacing this like squamous tissue? It actually is regenerating all the different epithelial cell types. And so here the colors are switched. We have keratin 13 lineage in green and the different cell type markers in magenta. And so KRT5 is a basal cell marker. And we see there's the hillock-derived basal cells are generated. Looking here at a ciliated tubulin, again, this is ciliated cells. So hillock lineage-derived ciliated cells. Here we have CC10, which stains secretory cells, and we see hillock-derived secretory cells. GNAT3 stains tough cells. We see tough cells derived from the hillock. PGP9 neuroendocrine cells, and they're also lineage labeled from the hillock. And lastly, BSND stains ionocytes, and they're also generated after injury from the hillock. So the hillocks regenerate all the different epithelial cell types after injury. And so hillock properties so far, it's stratified, it's squamous, it's on characteristic locations on the tracheal surface. They survive injury and regenerate the epithelia. But we wondered, is it just because these are stratified and the cells are protecting basal cells? Is that what's unique about the hillock? Or are the hillock basal cells also unique? Are they different? Could you put any basal cell underneath the stratified structure, and would it act the same way? And so to get at this, we used our P63 driver. And again, this labels all basal cells. Here they're labeled in green. And so we see all the basal cells here labeled in green, both in the hillock, as denoted by keratin 13, and outside the hillock that are keratin 13 negative. You see DAPI stain in blue. This is 3 days after labeling. And then so we chase this out and we see what would happen 3 weeks. Within 3 weeks, and this is quite surprising, I'll tell you why in a second. Within 3 weeks, we see that the basal cells in the hillock turn over. They differentiate into the hillock luminal cells. And this is actually in contrast to all the pseudostratified basal cells, which are still basal cells. Typically, the pseudostratified epithelium is pretty quiescent and doesn't turn over that quickly. It takes months. And so this was very surprising to see that hillock basal cells differentiate, turn over very quickly within the hillock. And this is at homeostasis, without an insult or injury. So this was turnover. We then also wondered, not only are they differentiating, could they also be proliferating? And so to get at this, we again use that mosaic mouse to label single cells. Here we use keratin 13 Cree-ER, our hillock lineage driver, with the mosaic mouse to identify specific hillock cells, sorry. And here we see our hillock. Acylated tubulin in yellow, the hillock here in keratin 13. If I take away the keratin 13, we can see the individual cells here. And you can see individual cell clones, and then in green and blue. And then we see other clones here in magenta. And there's actually 9 different cells with the same color, suggesting this clone size is 9. And you may think, oh, that's not that big. But by comparison to the pseudostratified epithelia, that's dramatically different. So if we look at clone size here on the y-axis, in pseudostratified epithelial basal cells, compared to the hillock, we see there's a drastic difference. The average clone size in pseudostratified epithelia is only 2.5, while when in the hillock is 7.5. And we see clones as large as 60 in the hillock. So this suggests that hillock basal cells are highly proliferative at homeostasis, unlike the rest of the pseudostratified epithelia. So this was evidence that suggests to us that hillock basal cells are different. But we wanted to more rigorously characterize those differences. So we wanted to do sequencing to get a better sense of how these basal cells are different. So we used our hillock lineage driver, keratin 13 Cree-ER, with an MTMG reporter, so that all hillock cells will be green, while all non-hillock cells will be red, tomato, magenta. We then fluorescence activated cell sorted for the basal cells and did bulk RNA sequencing of the pseudostratified basal cells and the hillock basal cells here in green. And then we looked at the gene expression differences. And so here we see a volcano plot looking at log P over fold change of the gene expression differences between the hillock basal cells in green and pseudostratified basal cells here in magenta. And the first thing we see is that there are differences. So that's important. And those that would be the genes that would be more enriched and pseudostratified would be here on the left side, while those the more enriched in the hillock will be on the right side. And I've already labeled some of the important genes here. And interestingly, these genes are involved in vitamin A or retinoic acid signal. So here's a vitamin A pathway. Vitamin A is an essential vitamin, right? We have to take it through our diet. And the active form of vitamin A is all trans-retinoic acid, the most potent form. What's interesting is that we see a number of these enzymes that are important for the formation of ATRA, all trans-retinoic acid, is enriched in pseudostratified epithelia, while one of the main enzymes for breaking down, catabolizing all trans-retinoic acid is enriched in the hillock cells. And this was very interesting, primarily because of historical context. So nearly 100 years ago now, this experiment showed that vitamin A deficiency is associated with squamous metaplasia. And so what these investigators did also in Boston, like many of us, they fed rats vitamin A deficient diet. And what they found was that many of the epithelia became squamous. And this is actually a panel from their paper looking at the tracheal epithelium. And it looks very squamous. Some may say very hillock-like, in that there's these flat squamous cells, and this is a stratified epithelium. So we wondered, this metabolic program, this retinoic acid metabolic program, the differences, this duality between pseudostratified and hillock epithelia, could we capitalize on this to enrich our hillock phenotype? And so what we found was that hillock basal cells show increased proliferation with retinoic acid inhibition. So to get at this, we again first use a genetic model using our hillock lineage driver, keratin 13 Cre-ER. We expressed a retinoic acid dominant negative receptor. So this prevents retinoic acid nuclear signaling and effectively is retinoic acid inhibition for all intents and purposes here. And what we found was compared to control without the retinoic acid receptor dominant negative, when we look at proliferation with EDU staining, that's here in green, we see that hillocks are proliferative after injury, as one would expect, but they're even more proliferative in the setting of retinoic acid inhibition, retinoic acid receptor dominant negative. So this is a short term, this is overnight exposure. We then want to look what happens more longer term with injury and regeneration. So we labeled hillocks with our keratin 13 Cre-ER LSLTD tomato. We waited a week for washout, and then we performed cryo injury, and then watched over 2 weeks for how the epithelia regenerated. We did this either with control media or with retinoic acid inhibition, specifically AGN 193109. And so this inhibits retinoic acid receptor signaling. And what we see here is pretty dramatic, that the area covered by the hillock is much different compared to control. We see much more resurfacing in the setting of retinoic acid inhibition compared to control, which is consistent with these findings with increased proliferation. And so this was with injury. We then wanted to revisit that study from about 100 years ago, giving mice, in this case, a vitamin A deficient diet. So we took mice, we either gave them regular chow or a vitamin A deficient diet, and then looked to see what happened to their airways. Here's control. We have acylia tubulin staining in magenta and keratin 13 staining in green. And so actually, what's interesting here is what we don't see. We don't see that there's random patches of keratin 13 forming on this vitamin A deficient trachea. We actually see what we would expect with the hillock, keratin 13 staining over the posterior membrane and over the cartilage ring in these characteristic hillock locations. And so what was surprising to us was that it looked like, and we can quantify this as well, looking at keratin 13 area over total area. Here on the y-axis, control compared to VAD, we had an increase in keratin 13 area, but only in characteristic hillock locations. So it was quite surprising to see that actually this vitamin A deficiency induced squamous metaplasia is actually hillock expansion, and that the hillock has increased proliferation and is growing in a setting of vitamin A deficiency. So we then wanted to see whether we could use this to our advantage to recapitulate the hillock in vitro. So when we take hillock basal cells and pseudostratified basal cells and grow them in culture and form air-liquid interface alleys, they actually look indistinguishable. They both form ciliate epithelia, generate all the airway epithelial cell types. And we wondered, with using this sensitivity to retinoic acid inhibition from all this previous data, could we recapitulate the hillock in vitro? So we used our keratin 13 Cre-ER MTMG mouse, where the hillock again is labeled in green and the pseudostratified epithelia is labeled in magenta here. We isolated the basal cells and then made ALI cultures in the setting of retinoic acid inhibition. And what we found was a pretty dramatic difference between those cells derived from hillocks compared to those derived from pseudostratified epithelia. And we see this stratification, this multi-layered ALI in the hillock-derived cells, which is much greater than that we see in the pseudostratified epithelia, reminiscent of the hillock, and also has injury-resistant properties, which I won't go through here. So effectively can recapitulate the hillock in a dish. And so far, what I've shown in the mouse airway is that hillocks can be defined by their location along the posterior membrane, cartilage rings, their structure, here being stratified squamous epithelia, expression of keratin 13 and other genes like DSG3, and importantly, their function. Their injury resistance, a variety of different injuries, their sensitivity to retinoic acid, and the regeneration of the airway epithelial surface. So with this description in mice, the next million-dollar question is, do hillocks exist in humans? And the short answer here is yes. And so to get at this, what we did is we explanted entire airways from samples that were not suitable for transplant, but donated graciously for research. And here we see, this is a human trachea with keratin 13 staining in brown. And you can see very similar, very reminiscent to what we see in the mouse trachea over cartilage rings, these patches of keratin 13. We can microdissect one of these regions and do cross-sectional staining. And we see this is also very reminiscent we see in the mouse. The stratification enriched keratin 13 region. So let's take a look at this a little bit closer. So here we have H&E of this human section. And similarly, a few slices away is the same section just in immunofluorescence. And so what's notable here, looking at just the H&E, we can see that there's these flat cells, these flat, very squamous looking like cells, as a pathologist would say, looks like reserve cell hyperplasia. As we go out of the hillock, we start seeing these other cells that look like they have little hairs, the ciliated cells, so interspersed ciliated cells, and then a more ciliated region. And then this becomes more apparent here with the immunofluorescence, with a ciliated tubulin here in magenta, keratin 13 in green, and DAPI staining nuclei in blue. And you can see this very highly enriched keratin 13 region here with these flat squamous cells that look stratified. We have this transition zone, which is actually very similar to the mouse, which I didn't mention before. And then we have a more ciliated region here. And so, as I mentioned, is this a hillock is the question. But hillock is based on not only gene expression and structure, but function. So the question is, do these regions have the same injury resistance and retinoic acid sensitivity function? So to be able to do this, we needed to be able to identify hillocks in live tissue. And so this is a challenge because we can't use genetic modification in people to identify hillocks. But what we could do was use some clever ways to do live staining and negative contrast. So here we stain ciliated cells using a lectin, WGA, and that just binds the sugars on the cilia. And we could then identify these aciliate patches, which are putative hillocks. And that's what you see outlined here in magenta. So we can microdissect those regions and adjacent ciliated cell regions, culture those basal cells, and then do one of our first tests, retinoic acid inhibition. See if the human hillock basal cells also have this retinoic acid sensitivity. And it turns out, which I'll show you in a second, that they do. The hillock basal cells form in vitro hillocks, while the basal cells derived from these ciliated cell patches form normal pseudostratified epithelia. And that's what you see here. The regions derived from hillock compared to those derived from ciliated cell. So in vitro cultures, you can see the hillocks are enriched in keratin 13, have no ciliated cells or aciliate in contrast to these pseudostratified patches. And they also demonstrate increased barrier function properties, one of them being transepithelial resistance, which is a measure of barrier function, essentially. That's what you see here down on the y-axis. In hillock cells compared to ciliated, ALI derived from hillocks compared to those derived from ciliated epithelium. But the million-dollar question then is, do these in vitro hillocks also survive injury? And so we did our battery of tests. We tested flu infection, cryo injury, and acid injury. And what we see here for hillock staining keratin 13 in magenta and flu NP in green, hillocks are protected while pseudostratified ciliated epithelia is not. And that's quantified here on the y-axis. hillocks, a lot less flu compared to the ciliated alloys. Similarly, for cryospray and acid injury, very few dead cells. There's a few dead cells here in both acid and cryospray, but much less by comparison to the pseudostratified, the ciliated epithelium. And that's quantified down here. Hillocks, in vitro human hillocks, are protected from both cryo injury and also looking at dead cells per millimeter squared. Hillock is compared to ciliated cells, also protected from acid injury. So these human in vitro hillocks have conserved functional properties to what we saw in mouse, in vivo, and ex vivo. But these are humans. As with everything with humans, it's always a little bit more complicated. And so one of the things we mentioned in mice is that keratin 13 is one of the defining gene expression characteristics of a hillock. That's not true for human. Not everything that's keratin positive is hillock in human airways. So this is a cross-section of a ciliated region, as you can see with ATUB, acetyltubulin in magenta here, keratin 13 in green, and DAPI nuclei in blue. And you see that there's some of these basal cells are super basal cells that highly express keratin 13. And this is what we suspect is found in a lot of single cell RNA sequencing of these keratin 13 rich super basal cells. How these are related to the hillock, that's something that we have to figure out, or if they're even related to the hillock. Furthermore, it's not only on this microscope level, cross-sectional level, but also on a whole mount level, we see differences in keratin 13 staining. So this was the trachea I showed you earlier, showing those characteristic keratin 13 patches over cartilage rings and over adjacent to the posterior membrane. But as we go, this is the right main stem bronchus. As we go down, here's the bronchus intermedius. Zooming in, we see these other patches of keratin 13 positive cells. This looks more like spots, like leopard print. How are these related to the hillock? And do they have similar properties? We don't know. That's another area that needs to be investigated. But the most important thing here is that we need, in order to determine what's a hillock and what's not a hillock, we need three things, right? We need the structure, the gene expression, but also the function. The functional properties are really important in the definition of a hillock. And so understanding what those two regions are, we don't know without all three of these characteristics. And this is an opportunity for both spatial sequencing and coupled with functional assays to help better characterize what regions are squamous metaplasia, what regions are hillock, and what regions are something else in between. Actually, based on this definition, we can now say that hillocks are more defined based on their structure, gene expression, and function compared to this kind of catch-all term of squamous metaplasia. As I mentioned, the next steps of this work and other work is to better understand the hillock throughout the airways. Here we've just looked at the large airways, mainly the trachea and main stem bronchi. And so looking at this both in mouse and human is one of the next steps. Furthermore, this mechanism of injury resistance is quite fascinating, how hillocks can survive essentially everything. And so defining this mechanism of injury resistance is also ongoing work. And kind of thinking a little bit broader and implications for disease, even more so than regeneration. What disease processes are hillocks involved in? And what could hillocks act as a precursor to cancer? Is the squamous cells we see, the squamous metaplasia, a precancerous lesion, or is that something different entirely? And so all of this is very, very interesting questions and thought-provoking questions that are some of the next phases of this work. I just wanted to also mention that this work doesn't stand on its own. There's a lot of work, previous literature that has come before it that served as an important foundation for a lot of these foundings. People have described there's a heterogeneous population of basal stem cells, some of them that are unipotent, that particularly are dedicated to squamous differentiation, and that they're located here along the dorsal epithelium. Furthermore, that there's some zones that proliferate more than others, and that these are aciliate. And so there's been a lot of work a lot of pieces of this, but I think the hillock seems to put a lot of this together to synergize a lot of these findings into what this is. And so the hillock kind of explains a lot of these findings from the literature. So in summary, main points that you should take away. Hillocks are squamous stratified structures with a dedicated and unique population of stem cells found at homeostasis in both mouse and in human airways. One of the important functions of hillocks is that they survive denuding injury, regenerate the epithelial surface, and they have a unique sensitivity to retinoic acid inhibition. And so this is the cartoon that you can have in mind for what the hillock does. So here we have our airway epithelial surface with all the different cell types, including the hillock. In the setting of denuding injury, very severe injury, these cells die, but the hillock survives. And hillock basal stem cells go out and expand and regenerate the airway epithelial surface, regenerating all the different airway epithelial cell types. Great. Lastly, I just wanted to thank everyone involved in this work, particularly Jay was a phenomenal mentor and great guiding light for all of this work. The other co-authors, my other co-first authors, Brian Lynn and Chaim Chernoff. This was truly a team effort, and it couldn't be done without everyone here, particularly the co-first authors. And I just wanted to mention, as I mentioned, we had a very large team that helped make this happen. From our lab and our collaborators, and particularly Lida, who really helped us put this in a pathological context as well. Wanted to thank the Division of Pulmonary Critical Care Medicine at MGH and the Center for Regenerative Medicine. And then our funding sources, NHLBI, CFF, LungMap, and of course, New England Donor Services for the gracious gift and access to human lung tissue and airway tissue to be able to identify the hillock in humans. Great. So I think we have some time. I'd love to have discussion and talk about some of this work.
Thanks so much, Viral. That was some really elegant work and also very elegantly presented, I have to say. As someone who does not do this on a day-to-day basis, it was still very easy to understand from the way you talked about it.
Appreciate it.
I wanted to open it up if any of the panelists had, a few comments that they wanted to say or any anything about how this maybe changes their mental framework about stem cells in the lung in general. Maybe Dr. Hariri, if you don't mind.
I was waiting for Jay to unmute and say something. I thought I'd give him first stab, but I will go first. I thought that this was super fascinating. I think from our perspective in pathology, it's so fascinating. Jay and I were like joking that I see these cells all the time. And we were like, oh, it's just like reserve cell metaplasia. Like they're just reacting to something. Like we sort of had this intuitive inkling about what these things were doing. But I never really thought of them as like a reserve cell population to be able to like respond to injury so quickly. But after all of this work that Viral and Brian and Jay have done that's just been so gorgeous, and being able to collaborate with them on that, I feel like it's totally changed my framework of how I think about the way that the lung repairs and how much smarter it is than I ever had thought. Before I thought it was, those little basal cells would just like rev up and replicate, but that's so slow, especially when you think about how easy it is to injure the airway and how hard you can injure the airway at any given moment. Like to have a little like army population like ready to go and then expand, like the way a lymph node has like, a clone that can expand is just so fascinating. And it was such a pleasure to get to work with them.
Thank you so much. Dr. Rajagopal, do you have any additional comments on top of that?
I mean, I think Viral did a great job of summarizing the main conclusions. I mean, from my point of view, now, this was a difficult paper to write because it was so confusing. It was the hardest thing I've ever had to pin down. And fortunately, the lineage tracing mouse was very useful, and demonstrating that these structures were permanent, not permanent, but lasted for months was useful because of this idea of just an injury response from the epithelium wasn't consistent with the fact that they would last for quite a long time. Plus, there was no injury because these were mice walking around in in a cage with no obvious injury. The other thing that I learned working with Lida especially is that the definitions of these terms like squamous metaplasia aren't as trivial as one might think. If you were going to write an article to define squamous metaplasia, I think you would have to describe like 10 different things, sometimes with markers, sometimes the markers aren't all consistent. So we tried very hard, as Viral showed, to come up with a very strict definition for hillock. And some of that may overlap with the term squamous metaplasia and some may not. But really, at the end of the day, I'm left with like far more questions than answers. Like, what does this have to do with cancer? When does this arise in injury? What explains these bizarre patterns? You know, it's not like a hand where everything is always in the same place. And so, yeah, I mean, that about summarizes it. There's lots and lots of work to do. Or, for example, the clonal expansion. How is one of these hillock cells able to like generate 10,000 cells? Is kind of an interesting, mysterious question in and of itself.
Mhm.
And I'd add on top of that, I think the link to cancer is so fascinating because I think for a long time we thought that like squamous dysplasia had to be the precursor to squamous cell carcinoma. But if that were to be true, we would expect every squamous cell carcinoma or nearly all of them to have a background of squamous dysplasia the way the cervical cancer does, right? Like cervical cancer, you never get a cancer without having a dysplasia present there. But in the lung, that's not true. And I think we always just were like, I don't really understand that. And now here, I think there's this potential of, as these cells are developing, can they develop a clone that goes haywire and then takes a squammy route? Who knows? And I think that's super fascinating. And can't wait to dig into that with these guys too.
Dr. Fine, and I noticed that you wrote that you had a question. Would you care to elaborate
Yeah,
on
thank you. That was a really beautiful talk. I was wondering whether you could provide any insight to the fact that these cells have very specific are observed in very specific locations. And then also secondly, you talked about a little bit, but if you could speculate on why these cells are resistant to a host of different kinds of injuries. Thank you.
Yeah. Fantastic questions. We've also been struggling with those questions as well. To answer your first one, why the hillocks arise where they do. And so there's clearly some sort of niche. And this answers another question in the chat as well. So I mentioned here that the hillocks are characteristic, but they're not stereotyped. So they're not in the same exact location, even in these mice that are very pure population. And so they can vary, being on one side of the trachea versus the other side on not every cartilage ring. And so we don't totally understand that quite yet. We suspect that there's some sort of niche factors. We suspect it's probably related to maybe the tension of the airway, Jay always says, is a mouse turning its head one certain way a lot? Could that be stimulating the hillock? And I think that's an important question that we don't understand. That also carries on to the human as well, since these are even more variable in the human from what we've seen. I think that answered your first question. Right, Alan?
Yes.
Okay.
But the second question had to do if you could speculate on the mechanisms of the resistance to all these different types of injuries.
Excellent. Yeah, excellent question. And it may not be one common mechanism. It could be. Actually, this is one of the main specific aims of my KO8 that I'm in the process of resubmitting. So it's one of the things we want to get to better understand. Could it be because they have these intermediate filaments, very enriched in keratin and desmoglein, these tight structures that provide a barrier similar to the skin? That could be one mechanism, how hillocks are protected against some of the injuries. Another mechanism could be that they don't have some of the enzymes, specifically for naphthalene toxicity. You need some of the important enzymes to make it the active toxin product, and they may not have that. So there could be multiple different mechanisms. We're actively trying to figure that out as well. It could be due to the unique structure of the hillock.
Thank you.
Right.
It
Let me
looks
have you
like-
weigh in very very quickly, Dan, if you don't mind, or Viral.
Yeah, go ahead. Yeah.
Please.
I think all those questions that you already covered, sort of that exist as your current future directions, are like definitely so exciting. Just to kind of take a departure, I guess, have you guys looked at any different developmental stages or embryonic development for the presence of these cells, where they exist, what they look like?
Yeah, a great question. Actually, my summer student right now is doing a lot of that work also. We haven't looked quite at embryonic stages yet, but a few days after birth, we start seeing these hillock patches rise. I think it's most clear like about P6 where we see this form. We're still doing it more rigorously, and particularly with lineage now that we have the mouse. But yeah, we do see these form pretty early on. But whether
cool.
they're there embryologically would be very interesting. And I don't know that answer yet. Right.
Okay, I'm trying to see. I think a lot of the chat questions have been answered already. There's a question about, are there any better surface cell markers in the human hillocks?
Yeah, the human's been a challenge. It's been a challenge, particularly because of the keratin 13 superbasal cells that are not in the hillock regions. And trying to understand that is important. We're in the process of doing spatial sequencing of human hillocks to better get those cell surface markers. We haven't found anything quite yet, but we're still in early days with that spatial technology.
Got it.
Yeah, wish I could give you a one, Scott.
And then there's another question about differences with age with the responsiveness of the hillocks, not necessarily the presence, as Erin asked.
Yeah, that's a great question. We haven't looked at the functional consequences as a mouse ages. What we have noted was that hillocks seem to get bigger as the mouse ages. But whether that has a functional consequence, we don't know. That's an interesting question that we should look into.
And then there's one more question about resistance to injury as being reminiscent of wound repair, where a mechanism is senescence of specific cells. Are there any differences in the senescence markers that you see in the hillocks, or have you looked at that?
Yeah, we haven't looked specifically at senescence, at least for this manuscript. We're trying to understand more of the proliferation and basal cell properties of the hillock in particular, and are there any unique cells in the hillock? But we haven't looked at senescence in particular. But that could be an interesting thought. Do they come in and out? And is there maybe a super hillock basal cell that could do that? That'd be very interesting.
And then I actually had a question from my perspective. It's kind of tempting to extrapolate some of these things to, for example, cigarette smoking as a chronic mechanism of injury to the airway epithelium, and then, the connection there to, squamous cell cancer. The other sort of thing that comes to mind would be something like ARDS and what is the role in this? And maybe they sort of play, in one context, they could be bad in terms of metaplasia, and in another context, they could be good in terms of regeneration. Obviously, that's very broad and outside the scope of your current work, but do you have any thoughts about those sorts of questions?
Yeah, I mean, like one of the thoughts, let me just show this slide at the end. One of the thoughts here is that could there multiple repeated injuries or specific toxins or something, and Lida mentioned this as well, cause some of the basal cells to be dysregulated and they can no longer differentiate, or they differentiate poorly. I think that's really interesting and a really interesting hypothesis. Could something go haywire? And again, it's like this duality, like you're saying. You want to cover the airway. It seems like there's maybe a different stages in airway injury and repair. You want to just cover everything so you're not exposed initially, and then you kind of pick up the pieces later. That could be one process that's going on. Or you could try to do it a little bit more, maybe intelligently with the right cell types, and maybe that's what the hillock is. But we don't know. I think that's all speculation, trying to figure out what that is. Yeah.
Okay, we're approaching the top of the hour. Are there any final questions? Oh, someone just popped up in the chat. Can you see that, Viral?
Yeah. Distinguishing between Car T13 superbasal cells and Hillock basal cells, do we know that the superbasal cells don't have the Hillock properties? Yeah, we don't know that. That's a great question. It would be very interesting if they have very similar properties in terms of proliferation and turnover. And I think that's something that we need to look into. Absolutely.
Okay.
I
Well,
could just say super quickly.
yeah.
Sorry, I was just thinking, Alan and I actually do lymphatic research, and so just the pattern that you see in the trachea is not dissimilar to where the lymphatics lie. I assume also, blood vessels and blood vasculature. So that's kind of I don't know if there's a connection there. Obviously, a couple layers away, but
Interesting. Yeah, maybe we need to do some staining for lymphatics. Yeah, thanks. It's a great idea.
Well, thank you so much. I think it was a really interesting presentation and a lot of great discussion about it. As always, it's always more complicated than we think. So
Yeah, absolutely. And if anyone has any other questions, feel free to reach out to any of us. Jay's contact information is on the paper. Our email's on the website, so feel free. We're happy to discuss anytime.
all right. Thank you so much, everyone. Take care.
Thank you.